Okie doke. Here we go. Chapter 16 Carbohydrates, Sugar Chemistry. The first job that I ever had working in a lab was working in a carbohydrate research group and learned a little bit about carbohydrates, but I was pretty young. So, anyhow, let's get started in talking about carbohydrates. It says carbohydrates are synthesized by photosynthesis in plants. This is where plants take water and carbon dioxide and they can make sugars like glucose. And sugars are found, or sugars are carbohydrates, are found in grains, cereals, bread. Of course, they're found in sugarcane. Glucose is a major energy source of the body. Your brain is using glucose as we speak. Otherwise, you'd be in serious trouble. It says here that a gram of digested carbohydrate gives about 4 kilocalories of energy. So not as much as a fat, and it's around the same as a protein, which also gives 4 kilocalories per gram that's digested. What else? Complex carbohydrates like the ones found in potatoes, those are best for our diet. And then the USDA recommends that about 45 to 65% of our daily calories should come from carbohydrates. I think that I'm guilty of, consuming more than 65% some days. So something you want to think about. Anyhow, there's some basic carbohydrate types that you all have to be aware of. And we're going to take a look at them on this slide, namely monosaccharides, disaccharides, oligosaccharides, and polysaccharides. Mono means one, and so it's not much of a stretch to realize that a monosaccharide is just a single carbohydrate molecule. Some examples of monosaccharides are things like glucose and fructose, and you could Google or just search, monosaccharides, and you come up with allose and mannose and aldose and all kinds of different monosaccharides. Glyceraldehyde is also a monosaccharide. But again, these are just one sugar, or what we call a saccharide molecule. A disaccharide, obviously, this contains two sugars linked together, or two monosaccharides linked together. For example, sucrose is a disaccharide that's formed from glucose and fructose, which are, again, both monosaccharides. Lactose is formed from glucose and galactose. And then maltose is formed from glucose and glucose. And we'll take a look at some examples of disaccharides like the ones that I mentioned. Oligosaccharides is the one that people, don't, oftentimes people will come in and they've heard of a mono or a disaccharide and even a polysaccharide, but maybe not an oligosacharide. When you have 3 to 10 monosaccharides linked together, that's what we call an oligosaccharide. It's basically a really small polysaccharide, because a polysaccharide, this is where we have huge, or bigger than 10, I guess, but really can be really big chains of monosaccharides linked together. And we're going to look at the polysaccharides that make up starch and glycogen, which are actually pretty closely related to each other. Starch and glycogen are. And then we'll also talk about cellulose, which is found in plants like grasses and wood, things like that. And again, those are just chains of monosaccharides all linked together. So monosaccharide, a disaccharide, oligosaccharide, and polysaccharide. Section 16.2 deals with monosaccharides. So monosaccharides are composed of mainly carbon, hydrogen, and oxygen. And they have this basic formula where we have one water molecule for every carbon atom. So if we take the formula CH2O and then we multiply that by N, where N could be any integer from 3 to 7, we end up with a monosaccharide. And so in case you never knew where the word carbohydrate comes from, you get the carbo here, right, from the carbon, and then the hydrate means for every carbon, we have a water molecule. So carbohydrate. Anyhow. All right. Many monosaccharides contain other chemical modifications or functional groups that don't involve carbon, hydrogen, and oxygen necessarily. Or I guess what I should have said is monosaccharides are not only composed of carbon, hydrogen, and oxygen, because in an amino group, of course, you have hydrogens in there, or phosphates have oxygens. But anyhow, we also see other atoms like nitrogens in an amino group, or phosphorus in a phosphate group. So we can have amino sugars, we can have phosphates on sugars. There's all kinds of variations that we can see on sugar molecules, and we'll look at a few of those in this chapter. How do we name a monosaccharide? Well, we name on the basis of the functional groups, and you can see that if we look at these two carbohydrates right here, there's you're like, wow, there's quite a few functional groups here. I see a lot of hydroxyls, and I see some carbonyls. For example, over here, we see that we have an aldehyde. And when we have an aldehyde carbonyl in our monosaccharide, we call that an aldose. And when we have a ketone carbonyl, because this carbonyl is flanked by two carbons, it's a ketone, we call that a ketose. So we can kind of divide our monosaccharides up into two really broad groups, okay? Ketoses and aldoses. When you see an aldehyde in the molecule, it's an aldose. When you see a ketone in the molecule, it's a ketose. Beyond that, you take a look at the number of the carbons in the skeleton. Okay, how many carbons are in the entire molecule? That's what it means by skeleton. Well, if you have 3 carbons in your monosaccharide, we call that a triose. If you have 4, we call it a tetrose. 5 is a pentose. 6 is a heptose. And you're wondering, well, what about a heptose or an octose? Yes, those are things for sure, but we're not going to look at any of those. We're mainly going to deal with trioses, tetroses, pentoses, and hexoses. And if we combine those two concepts together, we can provide even more information about the molecule. Let's go up to the examples that are on the top first. Okay, if we look at this molecule here, which says it's an aldose. Again, how do we know that this is an aldose? Because it has an aldehyde in it. Yes, there are these different hydroxyls here, and you might think, well, isn't it an alcohol? Well, there's something in organic chemistry called the order of precedence. And it's not something that we talk about in a lot of detail in this class, but I'll just tell you that an aldehyde takes higher precedence than an alcohol. So if you look at a molecule and it has an aldehyde functional group and a hydroxyl in it, you would call the molecule an aldehyde. You wouldn't call it an alcohol. The same thing applies for a ketone. Since we have a ketone in this molecule here, we call this a ketose. So an aldose and a ketose. Now let's look at these 3 molecules that are on the bottom, and you're gonna see these molecules come up not only in this chapter but in other chapters. We have this simple molecule here that's called D-glyceraldehyde. We have the next one, which is D-glucose, the most common monosaccharide on Earth, and then we have D-fructose over here. Now it's a little bit hard to see it based on my margin, but if we look at D-glyceraldehyde, okay, the first one. The first thing that I notice is that it has an aldehyde functional group. So that means it's an aldose. Then if we count up the carbons, starting at the top, 1, 2, 3, it's a triose. And we can put those two together and say it's an aldotriose. Let's look at glucose. It is also an aldose because it has an aldehyde on it. Right? It doesn't have a ketone. If we count the carbons starting at the top, we have 1, 2, 3, 4, 5, 6. And so not only is it an aldose, it is a hexose. We put those together and we make the word aldohexose. And then finally for fructose, you can see that this is a ketone. We have the ketone here. I'll try not to switch slides by accident. And if we tally up the number of carbons, starting at the top, we have 1, 2, 3, 4, 5, 6. So not only is it a ketose, it's a hexose, and then we can put those together and call it a ketohexose. The names that I have highlighted in yellow are their common names, but you don't have to memorize the structure of any sugars. If you want to memorize the structure of any of them, I would say it might not be a bad idea to memorize the structure of D-glucose just because it comes up so much in biochemistry. Section 16.3 deals with what are called stereoisomers in stereochemistry. You probably noticed on the previous slide that we said D-glyceraldehyde, D-glucose, and D-fructose. And you might have been wondering, what the heck do those D and L things mean anyway? The prefixes D and L in a monosaccharide name identify one of 2 isomeric forms. And these isomers differ in the spatial arrangement of atoms and are called stereoisomers. Notice the wording here, okay? Notice it very carefully that it says stereoisomers differ in the spatial arrangement of atoms. They don't differ in the way the atoms are connected. The atoms are connected in the exact same way, but the way that they are arranged in space is different. Stereochemistry is the study of the different spatial arrangement of atoms. We're not talking about constitutional isomers here. We're talking about molecules that are connected in the same way, but the way that the atoms are arranged in space is different. The stereoisomers DNL glyceraldehyde are non-superimposable mirror image molecules, and we give that a sassy name. We call them enantiomers. An enantiomer is a subset of stereoisomers. There are several types of stereoisomers. We're mainly going to look at enantiomers, and we'll look at one other kind, I think. But you need to know this definition that non-superimposable mirror image molecules are called enantiomers. Okay, you have to know the word enantiomer. Here's an example of two enantiomers. You see these two molecules here, and we have a mirror plane in between them, and you can see that they are perfect mirror images of each other. And the question I want to ask you is, are they the same molecule? And the answer is no, they are not the same molecule. If you were to try to take this molecule and overlap it on top of this one, the red and the red would line up, the green and the green would line up, but you see that this purple one right here, that would overlap with the yellow, so that wouldn't work. And then the yellow one would end up overlapping with the purple one. And if you're wondering, you know, Mr. Dion, isn't there a way I could manipulate this molecule to get them to overlap? The answer is no. There is no way that you can get these to overlap such that everything will overlap completely. Let me prove it to you. Here are two mirror images, two molecules that are mirror images that I've made, okay? If I try to overlap these molecules on top of each other, you can see that the two green spheres line up, as do the orange, but the orange and the white do not. Now you might be wondering, well, is there a way that you could manipulate those, or couldn't you twist them around? I think maybe if you put them this way, then they're going to overlap. Yeah, that'll work. The answer is no, it won't, because if you try, you can see that the green and the yellow will overlap. Oops, you can see the green and the yellow overlap like that, but the white and the orange, the white and the orange will not overlap. So the answer is these are completely different molecules. They differ in the arrangement of atoms in space. The black sphere in the center is connected to a green, an orange, and a yellow. In both cases, the connectivity is identical in these molecules. Okay, connectivity is the same. It's the arrangement of the atoms in space. Another example of that would be your two hands, okay? My hands are mirror images of each other, but are they identical? And the answer is no, they are non-superimposable mirror images. I cannot put my left hand in a right-handed glove. And that's what's shown at the bottom of this slide. You see the two human hands, which are in fact mirror images of each other, but we know that our hands are not identical. So to recap, the definition of enantiomers is non-superimposable mirror images, which I guess is already on this slide, but it helps you to memorize things when you write them down. Okay, so non-superimposable mirror images, that is the definition of enantiomer. Chirality, not pronounced chirality, it is pronounced chirality. A carbon that has 4 different groups bonded to it is called a chiral carbon. Remember that carbon, or that model that I put up a few minutes ago? It had a black sphere in the middle. We'll say that's a carbon. If that carbon is bonded to 4 different groups, I'll draw the tetrahedron like this, okay? Let's say you have, a chlorine here. Let's say you have a fluorine here. Let's say you have, I don't know what else. You could have an iodine, and let's say you have a bromine here, okay? That carbon is attached to 4 different atoms. Now, bear in mind, it doesn't say that the carbon atom has to be attached to 4 different atoms. It says it has to be attached to 4 different groups. Let me give an example of what that means. If you replace that bromine with a methyl, okay, a CH3, the carbon is still attached to 4 different groups. A chlorine is not the same as a fluorine. A fluorine is not the same as an iodine, and a methyl group is not the same as a chlorine, fluorine, or an iodine. And so this carbon here is a chiral carbon. Chiral carbon. Any molecule containing a chiral carbon, even just one, can exist as a pair of enantiomers, non-superimposable mirror images. The chirality in that little tiny molecule that we looked at earlier on glyceraldehyde, and we'll look at it again, okay, is conveyed by a chiral carbon. In fact, let me draw you the structure of D-glyceraldehyde. I'll draw it right here. We have a carbon. It's attached to an aldehyde. So we have an aldehyde up here. We have a hydroxyl. We have a CH2OH, and we have a hydrogen. This carbon in the center, this very carbon right here, is a chiral carbon. It is, in fact, chiral. Why? Because that carbon is attached to 4 different groups, not 4 different atoms, 4 different groups. Watch carefully. A hydrogen is not the same as an aldehyde. And hydroxyl isn't the same as an aldehyde or a hydrogen. And CH2OH is not the same as any of the other 3. And thus, this carbon is a chiral carbon. Larger biomolecules can often have more than 1 chiral carbon. In fact, if you look at a molecule like cholesterol, it has, I can't remember exactly, but I think it's like 8 stereocenters, 8 chiral carbons, which are also called chirality centers or stereocenters. Let's get back to that glyceraldehyde molecule. Glyceraldehyde has that chiral carbon. Remember, the chiral carbon in glyceraldehyde, I'll highlight chiral carbon, is this carbon in the center. It's got 2 enantiomers. You have the D isomer on the left and the L isomer on the right. How do you differentiate between the 2 of these? Because if you look at them, they both have an aldehyde on the top. They both have CH2OH on the bottom, but where they differ is the placement of the hydroxyl and the hydrogen. You can see that in the D isomer, or D-glyceraldehyde, the hydroxyl at the stereocenter is on the right. And that's how we know that we're dealing with a D isomer. It's when the hydroxyl on the stereocenter is on the right. When we're dealing with the L stereoisomer, the hydroxyl on the stereocenter is going to be on the left. Now, the next question to ask, and it would be a very good question, would be, what about, if I have a bunch of chiral centers, in a molecule? Well, we'll get to that in a second. Let's look at a ball and stick model of glyceraldehyde. This is just a molecular model. You can imagine building these in the lab. You all know how to make double bonds and single bonds. You can see the 3 carbons in glyceraldehyde. You can see the aldehyde at the top. You can see the CH2OH down here, CH2OH. Boy, how do they differ? They differ in the placement of the OH in the D-glyceraldehyde is on the right-hand side, and in L-glyceraldehyde, that hydroxyl is on the left-hand side, like that. Let me just address one more thing with you before we move on. If you're wondering, hey, there's 3 carbons in the molecule. Aren't the other 2 carbons, aren't they chiral carbons? First, I want to look at this carbon right here, the carbon number 3 in both of these molecules. Let me just ask my students who are listening right now on Microsoft Teams. Are either of these carbons that I have circled in blue, are these chiral carbons? Yes or no? Yeah, okay, I'm getting a resounding no from my students, and you're all correct, right? It's impossible for a CH2 to be chiral because it's got 2 hydrogens attached to it. If you've got 2 atoms that are the same, there's no way that that carbon could be attached to 4 different groups. And so the carbons that are in blue, these are not chiral. Okay, let's look at the 2 carbons that are labeled carbon number 1. I'll show you, I'll do this one for you, but this carbon is only attached to 3 groups. In order for a carbon to be chiral, it's got to be attached to 4 different groups. This carbon is only attached to the oxygen by a double bond, a hydrogen, and all of the rest of the molecule. So that is not a chiral carbon. It does not have 4 different groups, and this carbon is identical. Okay, there we go. So now we know that the 2 carbons that are circled in red are not chiral, and the 2 carbons that are circled in blue are not chiral. In fact, if you want a quick and dirty rule, you might say, hey, Mr. Dion, I see this a lot in organic chemistry. I see CH2 a lot in organic chemistry. That could never be chiral, right? You would be 100% correct. That could never be chiral The same thing, what about a methyl group? Could the carbon in a methyl group ever be chiral? The answer is no, because it's attached to 3 groups that are identical, right? So we could even put here, can never be chiral. All right. Now I want to talk to you about what's called optical activity. And you probably remember that at the very beginning of the lecture, I said, okay, if we have 2 enantiomers, they're going to have the same physical properties, right? If I go back here and look at glyceraldehyde, DNL glyceraldehyde, they're going to have the same polarities, right? All the bonds are in the same places. They're just mirror images. They're going to have the same melting points, the same densities, the same boiling points. How would we differentiate between the two? If somebody gave you a bottle with L-glyceraldehyde in it and somebody gave you a bottle with D-glyceraldehyde in it, how would you be able to distinguish between the two of them? And there is a way that we can do that. And it's based on the concept of optical activity. Now, you heard the word optical, you think, hey, this must be something that I could see. And you would be 100% correct in assuming that. Here's what optical activity is. Enantiomers are also sometimes called optical isomers. What they do is that they interact with plane polarized light and they rotate the light in opposite directions. And we call this optical activity. So to summarize what it's saying here, it's saying that if we take light and we polarize it, we have one plane of light. And if we shine that polarized light, that one plane of light, through a solution of an optical isomer, it's going to rotate the light in one direction. If we have the enantiomer of that, it's going to rotate it in the opposite direction. And this is how we distinguish between optical isomers or enantiomers. And the device that we use is called a polarimeter. Let me show you what a polarimeter looks like in a second. Let me give you some more information on plane polarized light before I show you the polarimeter. Okay, if we have normal light coming out of just your regular, run-of-the-mill, pretty good drawing, isn't it? If we have a run-of-the-mill light bulb, looks like this, it's got a filament in it, okay? Well, that light bulb is going to shine, it's going to shine light, okay? And it's going to shine it just in all directions. So that light that comes out of that light bulb vibrates in an infinite number of directions perpendicular to the plane of travel. But when the light passes through what's called a polarizing filter, this is the same kind of polarizing filter that you would find in, I don't know why it says Polaroid sunglasses, it should say polarized sunglasses. Only the light vibrating in one plane is going to reach the other side of the filter. And then the polarimeter allows the determination of the specific rotation of a compound, of a chiral compound, and it measures the ability of the compound to rotate plane polarized light. Look, a picture is worth 1,000 words, so let me show you what the polarimeter looks like. Here's your light source on the far left. Okay, here's our light bulb. Now, when we shine light, again, it vibrates in all directions perpendicular to the plane of travel, right? The plane of travel of the light is this direction. And you can see that the light is going in all directions, right? Every single direction, even the ones in infinite number of directions. So when you pass it through a polarizing filter, it says polarized filter. When you pass it through that polarizing filter, all it does is it allows one direction of the light to come through. So you saw all those different directions of the unpolarized. The only one it's going to let through is the one that's going to fit through the slits, if you will, in the polarized filter. So now you have plain polarized light. You take your organic compound, okay? You take a chiral compound, for example, okay, or one enantiomer, and you make a solution of that in some kind of organic solvent, okay? And then what happens is the plane polarized light is actually going to interact with the molecules that are in here. Okay, remember it's a solution, so the light is actually going to be deflected by the light will be deflected by the molecules that are in there. Now you can see that it's rotating this plane of light. It's rotating it in this direction, right? It's rotating it down here. That alpha, this is the angle of optical rotation. And what you really need to know in Chemistry 102 is this, okay? So pay close attention to this, is that if you have enantiomer A, let's say, we'll say enantiomer A, and it rotates plane polarized light, let's say in this direction by, I don't know, let's say it's arbitrarily, let's say it's 20 degrees that it rotates the plane polarized light. If you have enantiomer B, and you measure its ability to rotate plane polarized light, it's going to rotate at 20 degrees, but in the opposite direction. Okay, so if this was negative 20, this is going to be positive 20 degrees. Okay, that's the main thing that you need to know in this class, is that enantiomers are going to rotate plane polarized light in opposite directions, but the same amount. Now, I have a question for you guys. What if I took a 50-50 mixture of enantiomer A and enantiomer B, I mixed them up, and I put them in here, and I shone the plane polarized light on a 50-50 mixture of enantiomer A and enantiomer B? What do you think the optical rotation would be? So if it was 180, that would mean that it would rotate it all the way, and you'd have to spin it down here. Okay, the answer is 0. Yeah, Greg, you're 100% correct. It's not going to rotate it at all, because enantiomer A would rotate it, let's say, what did I say before, negative 20 degrees, and enantiomer B is going to rotate it positive 20 degrees, and what are they going to do? They're going to cancel each other out, and so you get no optical rotation. If you're thinking, well, that's not very helpful, well, not really, okay? But it is a way for us to distinguish between pure samples of enantiomers. Give me a thumbs up if you follow me on just the concept that those two enantiomers, those non-superimposable mirror images, are going to rotate the plane polarized light in equal but opposite directions, okay? So let's say you pick any number. Let's say one rotates plane polarized light negative 37 degrees. The other one's going to rotate it positive 37 degrees. Let me just tell you one more thing before we move on. And I can't remember if I have this in my slides or not, but this is just something that you might find fascinating. You know what? Let's go over to the next slide first, and then I'll tell you. I'll tell you what I was going to say. This slide just kind of summarizes the relationship between molecular structure and optical activity. So it probably covers a lot of what I said already. Let's go through it as a group. The first bullet point says when an enantiomer in a solution is placed in the polarimeter, the plane of rotation of the polarized light is rotated. One enantiomer will always rotate it clockwise, so that's positive. That's what we call the dextrorotatory. It's a pretty fancy word, but anyhow, I always think of French. The right hand, the right side is droite in French, so it starts with the letter D. And the other isomer rotates the light in the counterclockwise or negative direction. That's called the levorotatory isomer. Now, again, this is what I told you, is that under identical conditions, the enantiomers always rotate light exactly to the same degree, but in opposite directions. I can't stress this enough that you have to know this point right here. The thing that I wanted to tell you is this. If we back up, I'm going to back up quite a ways, but bear with me. I think you'll find this interesting. Let's look at these two enantiomers. Okay, this is the first two enantiomers, molecular enantiomers, not the human hand, but anyhow, these are the first two molecular enantiomers that I introduced you guys to this morning, right? D-glyceraldehyde and L-glyceraldehyde. If you have two enantiomers, let's say you have two sugars and they're both D sugars. Do you remember earlier on we looked at D-glyceraldehyde, D-fructose, and D-glucose? Let's say if D-glyceraldehyde rotates plane polarized light to the right, okay, it has no bearing on whether D-fructose, D-glucose, D-mannose, D-allose, D-xylose, any other sugar is going to rotate it to the right side, okay? So the D doesn't necessarily mean that the molecule is going to rotate plane polarized light to the right side, okay? It's kind of a funny thing to think about. You're like, what? Well, then why would they use that? Well, what you're going to see in a little bit is that D, again, only refers to the side that the hydroxyl is on. And it's always the hydroxyl on the last chiral carbon. If you don't follow me 100%, don't stress about it because we're going to look at it more. I just like to sometimes introduce things to my students a little bit ahead of time, give them something to think about while we're, moving through the content. Now, if you go back to, and I'm not asking you to rifle through your slides while I'm lecturing, but if you go back to something like glucose, you might have noticed, or you might even be thinking, like, holy smokes, glucose has a bunch of chiral carbons. Right? It's got a bunch of chiral carbons in it. How would I keep track of all those chiral carbons? Well, when a scientist named Emil Fischer was naming and characterizing monosaccharides a long time ago, he needed a quick way to write the molecules and to include all those chiral carbons and to give them the correct designation. Okay, and or a designation. Another way that I could say that is to say to orient the atoms in the correct way in space. And since there are so many chiral centers in these molecules, he devised what are called Fischer projection formulas, which are really quick ways of writing out chiral carbons. Okay, so the Fischer projection, I'm starting at the top, uses lines crossing through a chiral carbon to represent bonds. The horizontal lines on the Fischer projection, so these are two Fischer projections right here. Okay? The horizontal lines, okay, those are projecting out of the page, right? You see here they're wedges, and you know that wedges are coming out. As my students, the first class I ever taught in America, a long time ago, the student said, "Oh yeah, the Fischer projection. It's like the bonds that are horizontal. It's like they're coming to hug you." Okay. Hey, if that helps you remember it, no problem with this guy. The bonds that are vertical, like this bond and this bond, those are going behind the page, right? You can see here that they are represented by wedges. But again, to simplify things, we just cross like this where this carbon is, and that is what we call a Fischer projection. So when you see this projection, you know that these bonds are going behind the page, and you know that these bonds are coming towards you. And that's the Fischer projection. So it avoids having to draw countless, wedges and dashes, which are fine for small molecules. I use them all the time. But when you get into really big molecules with lots of chiral centers, they can get pretty painful. And in fact, they get very cumbersome and very difficult to decipher. Now let's look at those three monosaccharides that we started out with in the beginning of this evening: D-glyceraldehyde, D-glucose, and D-fructose. D-glyceraldehyde, we said there's only one chiral carbon. It's this one here, and we discussed that at length. I'm just going to throw this out to my students. Could anybody tell me, and it's not an easy question, but I'll see if anybody can answer it, how many chiral carbons are there in D-glucose? How many chiral carbons do you think there are, based off of what I've told you? Okay, I'm getting some different answers. Yeah, okay, I'm getting some different answers. The answer is actually, well, let's see if we can figure it out. Is the first carbon, is that a chiral carbon, yes or no? I'll answer that for you. The answer is no. And we already decided that this carbon, carbon number 1, is only attached to 3 things, right? It's attached to the oxygen, the hydrogen, and then it's attached to all the rest of the molecule like that. So that's 3 things, and so that cannot be a chiral carbon. What about carbon number 2? Is that a chiral carbon? Yes, it is. Let me prove it to you. It's attached to this aldehyde, this hydroxyl, this hydrogen, and this whole dang group down here. Those are 4 different groups. You can see that what's in these 4 different red circles, or 4 red circles, is all different. And so carbon number 2 is a chiral carbon. Now what we're going to do is we'll denote it as being chiral by putting a little star by it. Okay, so that's a chiral carbon. Now we move on to carbon number 3. What's it attached to? It's attached to a hydroxyl, a hydrogen, all of this up here, and all of this down here. Again, you can clearly see that what's in my 4 red circles are different. Remember, don't fall into the trap of saying, well, this carbon that I have highlighted in yellow is attached to 2 carbons. It's not chiral. It's not being attached to 4 different carbons. 4 different atoms. It's 4 different groups. We're always looking at the entire group. And so you can clearly see that carbon number 3 is, in fact, a chiral carbon. And we'll denote that with a little star. Okay, if we look at the 4th carbon, hydrogen, oxygen, this group, and this group, therefore it's chiral. And then the 5th carbon is attached to a hydrogen, hydroxyl, CH2OH, and all of this spinach right here. And so that is definitely a chiral carbon. The CH2 at the end, we already decided that that cannot be chiral because it's attached to 2 hydrogens. And therefore, the answer is D-glucose has 4 chiral carbons. In D-fructose, we only have 3. We have one here, and one here. Don't believe me? Try circling the groups, and you'll see for yourself that D-fructose has 3 chiral carbons. Now, the reason I have this slide in here is because I want to address where the name D comes from. I told you that D doesn't necessarily mean that all of these molecules are going to rotate plane polarized light in the same direction. What the D stands for is this hydroxyl that's highlighted in pink in each one of these molecules. And if you start on the top carbon, 1, 1, 1. When you go down, going from 1 to 2 to 3, so on and so forth, the last chiral carbon in the molecule, the last chiral carbon in glyceraldehyde is this carbon right here. The last chiral carbon in D-glucose is this carbon here, and the last chiral carbon in fructose is this carbon here. What do these 3 carbons that I have in a red circle have in common? The hydroxyl on all of them is on the right-hand side, and that is where the D comes from. Whenever you look at any monosaccharide, even if it's one you've never seen before, it's something you don't recognize, if you look at the last chiral carbon and that hydroxyl is on the right-hand side, bingo, you've done it. You've identified a D sugar or a D monosaccharide. If you look at a monosaccharide and the hydroxyl is on the last chiral carbon is on the left-hand side, in that case, you would have an L monosaccharide. So D and L monosaccharides. And if you're wondering, why are you spending so much time talking about D monosaccharides? Why aren't you talking about L monosaccharides very much? I see you're really hooked on D monosaccharides. The reason why is because the D sugars are the naturally occurring sugars. D glucose is the sugar is natural. Okay, it's produced from photosynthesis. D fructose, for example, is found in is fruit sugar, so on and so forth. Again, the L sugars usually are made in the laboratory. Those would be synthetic sugars. Let's get back to those compounds with chiral centers. We just spent quite a bit of time identifying chiral carbons. We need to cover a couple more definitions before we can move on. The first one is a racemic mixture, And do you remember before when we were looking at the polarimeter, I said, what if you had, an equal amount, if you had 50% of enantiomer A, and you use a capital and it looks like electronegativity. So if you had 50% of enantiomer A, and if you had 50% of enantiomer B, what you guys said is that there would be no optical rotation. They would be optically inactive. This means that their angle of rotation would be 0 degrees. That, again, is when we have equal amounts, a 50-50 mixture of a pair of enantiomers. This racemic mixture, sometimes we can call it a racemate. The next definition that I want you to be aware of, and this one's a little more complicated, is diastereomers. It would probably just do you wise to memorize this definition that diastereomers are a pair of stereoisomers having 2 or more chiral centers that are not enantiomers. So another way you could call diastereomers is molecules that are not superimposable. So you can put it here, not superimposable, and not mirror images. So if you have 2 molecules, they're not superimposable, but they're not mirror images. Mirror images. There we go. Gotta complete the thought. Then you have diastereomers. All right. The next one is what's called a meso compound. A meso compound is a compound that does have 2 or more chiral carbons in it, but there's going to be an internal plane of symmetry that causes it to be optically inactive. Greg asked a very, very, very good question, which is beyond the scope of our class, but I'll tell you, Greg, if you want to look it up, You can totally do that. What Greg asked me is this. He said, what if you had an unbalanced mixture of two enantiomers? What if you had 60% of enantiomer A, right, and you had 40% of enantiomer B? Could you get any information for that from that whatsoever? Well, you can see that enantiomer A is in excess of enantiomer B by 20%, right? There's 20% more of enantiomer A. And so we'd say that this mixture has a 20% EE, enantiomeric excess. Again, that's beyond the scope of our class, but you can get some interesting information from enantiomeric excesses. First time a student's ever asked me that, very good question. Okay, so it's called enantiomeric excess, and if you feel like looking it up, by all means do so, but it's beyond the scope of this class for sure. Let me just show you what a meso compound would look like before we move on. An example of a meso compound would be something like this. Okay, if I had a cyclohexane and I put a wedge with a hydroxyl on it like this, and if I put a wedge with a hydroxyl on it like this, okay? So I would have a proton going down here and a proton going down here. Now, here's what I want to do with you. I want to examine, with my red pen, this carbon right here. Is that a chiral carbon? I'll help you out. The answer is yes. This carbon that I have highlighted is in red is chiral. Look, it's attached to a hydroxyl, a hydrogen, a carbon with an OH and an H on it, and a CH2 over here. You can clearly see it's attached to 4 different groups. And so that carbon is definitely a chiral carbon. Okay, so we could put a little star by it and say, that's a chiral carbon. The carbon, what about this carbon here? I'll highlight it in blue. Is that a chiral carbon? The answer is yes. Yes, it is. Look, it's got a hydroxyl, a hydrogen, this carbon with a hydroxyl and a CH2. So clearly it's attached to 4 different things, 4 different groups. So that's a chiral carbon. This molecule has 2 chiral carbons in it. The rest are all CH2s, so those can't be chiral. But what else does it have in it? It has a plane of symmetry running through the molecule. And so since you can see that these 2 chiral carbons are mirror images of each other. And so when you make a solution of this molecule and you shine plane polarized light through it, yes, this chiral carbon is going to rotate the plane polarized light in one direction. Sure it will. It's chiral. Every chiral carbon can interact with plane polarized light. But the problem is you have the exact same amount of the other chiral carbon. And so it's going to rotate plane polarized light in the equal and opposite direction. So if you're thinking, hey, is a meso compound kind of like having a racemic mixture, but they're in the same molecule? Well, I shouldn't phrase it like that because that wouldn't be terminologically correct. But yeah, it might be something like that. Anyhow, so that's what a meso compound is. If you're just dying to know what a meso compound is. All right, let's get back to that DNL system. And this is something that I already went over with you. And here's in slide form, monosaccharides. We like to draw those in Fischer projections. Why? Because glyceraldehyde, not too bad, only 1 chiral carbon. So using the old dashes and wedges down here, not too bad, not too bad. We can just draw a Fischer projection where we just draw the cross like that. But what if you had something like glucose? Glucose has a bunch of chiral carbons. It's got 4 chiral carbons. That's a lot of dashes and a lot of wedges to start drawing with a pencil, okay? When we draw a Fischer projection, we always put the most oxidized carbon closest to the top. So that means your aldehyde for an aldose or your ketone for a ketose. You always put those pointing up towards the top vertically. The carbons are always numbered from the top. I showed you how to do that with glucose and fructose and glyceraldehyde, for Pete's sake. If the chiral carbon with the highest number has the hydroxyl on the right, it's a D sugar. If the hydroxyl on the chiral carbon with the highest number is on the left, it's L. And again, most common or natural sugars are D sugars, okay? And if you want to get frustrated, which I don't want you to be, of course, but let me just put this bug in your ear that later on we're going to discuss amino acids in a lot of detail in chapter 18. And the natural amino acids are the L amino acids. So it's kind of the natural sugars are the D sugars and the natural amino acids are the L amino acids. Just something to think about before we get there. Anyhow, we see the structure of D-glyceraldehyde. We already looked at that. Here is the chiral carbon. with the highest number? And you're like, well, there's only one carbon in glyceraldehyde anyway. Yeah, you're right. Okay. Well, here it is. You can see the hydroxyls on the right-hand side. And so we call that D-glyceraldehyde. When it's on the left-hand side, it's called L-glyceraldehyde. Nothing more than that. Okay? Nothing more than that. Give me a thumbs up if you follow me on DNL. Little dextrorotatory and levorotatory. All right, good.